I would like to ask everyone physically joining us in this room to raise up for 1 minute of silence for the horrible event that happened yesterday in Azerbaijan. Unfortunately, there was an aircraft crash, and there were 14, I think 13, 14 soldiers lost their lives. So please raise up for 1 minute of silence to give respect to them. Thank you very much. You may be seated. Okay, thank you very much one more time for joining us today on the second day of the sessions. We are honored to have the first speaker, Gözde Demirer, joining us online from California. Gözde actually received her PhD recently. So she actually graduated from Koç University in Istanbul in 2015. Then she received her PhD from UC Berkeley. And soon she's going to join to Caltech, and she will have her own research laboratory. And today, Dr. Demirer is going to talk about the development of nano and biotechnologies for crop genome engineering. Dear Gözde, if you are ready, please share your screen with us. Thank you very much for joining us.
Great, thank you. Let me get this set up. Sorry, not the screen. Are you seeing the correct view, or should I swap this place?
This is fine. You can proceed right now.
Okay. Okay, and can you hear me well as well?
We can hear you.
Okay. I think the connection is a bit slow, but
Can you hear us? We can hear you. You can come.
Okay. Okay. Yeah. Thank you, Emre, for the introduction, and I would like to thank all the organizers for inviting me to give this talk today at this workshop. I was going to start with a brief introduction of myself, but Emre has done that already for me, so I'll just skip to the talk outline. So today I'll be talking first about my PhD work, which was nanomaterial-enabled biomolecule delivery for plant genetic engineering. And in the remaining time, I will talk about my current postdoc work, which is tomato genome editing for increased nitrogen use efficiency. So I would I would like to start by describing my PhD research. During my PhD, I worked on developing nanotechnologies to deliver genetic cargoes and perform plant gene editing for food security and sustainable agriculture. So I've done this research in the lab of Professor Marquita Landry at UC Berkeley, and I was involved with multiple projects during my PhD. But for this workshop, I'm going to focus on the two projects. First one is carbon nanoparticle development for gene delivery, and how we use this platform for the CRISPR-Cas9 gene editing in plants. So as you may all be aware and interested in this area as well, plant genetic engineering can address a lot of critical global problems that we are facing, such as in the field of food industry. We can create disease and climate resistant crops using plant genetic engineering. Similarly, engineered plants can provide advanced biofuels, as well as a scalable way of synthesizing therapeutics. And lastly, we can use the plants and engineer them for the remediation of our environment and sustainability efforts. I myself are most interested in the food and the environmental applications of plant genetic engineering. Even though all of these promise and excitement, there are still some challenges in plant genetic engineering. And one of the challenges that I have focused is the biomolecule delivery into plants. And the tools for biomolecule delivery and transformation in plants are very limited. And that's mostly because of the cell wall that's around these plant cells that acts as a physical barrier for the cargo delivery into the cells. So here I'm showing the size exclusion limits of certain barriers around the plant cells. So if you look at the lipid cell membrane, these membranes are able to transport across anything that is smaller than 1,000 to 800 nanometer. But when we look at the cell wall, we see that it can only pass molecules that are smaller than 20 nanometers passively. So which was a main barrier of delivering particles inside the plant cells. So given this limitation, the most commonly used method in plant gene delivery is the bacteria, Agrobacterium, which many of us are using to do transformations in plants. This method is able to deliver DNA, but to a limited plant host species. So our crop of interest may not be transformable with Agrobacteria. And it can only deliver DNA cargoes. So we are unable to deliver molecules like RNA and protein. And that DNA cargo that we deliver gets inserted somewhere randomly in the plant genome that causes a lot of regulatory restrictions. So the current GMO crops we have in the market today, in average, took 13 years of research, and it had a very high cost of 136 millions. And around 40 millions of this cost was due to the regulatory pipeline. So when I started my PhD, motivated by the impact of plant gene editing and the lack of availability of delivery tools, I wanted to develop a plant delivery technology that can work in a plant species independent way and efficiently so that we could use it in many different crops, as well as we wanted to design it so that it can have less regulatory burden by making it abiotic and non-integrating. So to achieve this, I used nanoparticles, and nanoparticles have been used in many animal and mammalian systems for gene delivery. And recently, as we all know, we have used it for COVID vaccine delivery using the lipid nanoparticles. So in this project, I used a material called single-walled carbon nanotubes. So this is a material based solely on carbon. And in the center, we have this one called SWINT. So that's single-walled carbon nanotubes. And the reason why we wanted to use this is that if you look at its diameter, it has 1 nanometer in diameter. So it's very small in this dimension that could enter through the cell walls, but it's also very long. So this is a sphere. It's a cylinder that's hollow in the inside, but very long. So this means that it has a very large surface area that can load a lot of molecules like plasmids and proteins on the surface. When we compare this single-walled carbon nanotube, for example, to other carbon materials like this buckyball, which is just a sphere of carbon, it is 1 nanometer in every dimension. So this one is also very small, but it has a small surface area, so we can't deliver enough proteins or DNA using this type of a material. And when we look at molecules like graphene, that has a very high surface area, but it's also very large in each dimension, so it will be hard to enter the plant cells. Given these abilities, as well as its high stiffness, we decided to use single-walled carbon nanotubes to deliver genes into plant cells. So how this works is that for the initial development of this system, we wanted to load these carbon nanotubes with a plasmid that is going to express a Gfp gene so we can easily detect it. So we used a very simple plasmid that's driving a Gfp expression with a common 35S promoter. And we introduce these into the leaves of plants. So here we are looking at a Nicotiana benthamiana tobacco plant leaf, and it's injected with a needleless syringe, just like we do agro infiltrations. And once it's inside the plant tissues, we would expect it to be able to transport across the cell wall and the plasma membrane. And once it reaches the nucleus, this plasmid DNA will just make mRNA and the protein of interest, so in this case, GFP. Before looking at the protein expression, we wanted to see if these DNA-loaded carbon nanotubes can enter the plant cells. And for that, we started looking at the protoplasts. So protoplasts are leaf cells that we can extract from the leaves and remove their cell wall using enzymes. So this is an initial system that we wanted to test. Given it doesn't have a cell wall, it's an easier place to begin with because the nanoparticles would only need to pass through the cell membrane in this case. So here we are looking at an extracted protoplast here on the left, and we stained its nucleus using the stain DAPI that's shown in blue, and then we have the DNA labeled with Cy3, which is a red fluorophore. So in this case, we are delivering the free DNA without carbon nanotubes, and in this case, we don't see any internalization of this DNA into the protoplast. When we deliver the same DNA with carbon nanotubes, then we see that the DNA localizes both in the nucleus and the cytosol of this cell. So this shows us that the DNA carbon nanotubes can enter the plant cells when they don't have the cell walls. So next, we looked at if they could also enter into the insect leaf cells. And in this case, I'm showing here an image from a tobacco plant, and this is just a reminder that when we look at the epidermal cells of these plants, there is a huge vacuole in the center of this cell. So then the cytosol is pushed to the cell wall. So here the green contour is where the cytosol is, and this circle here is the nucleus. So keeping this in mind, when we look at the DNA carbon nanotubes that are tagged again with the Cy3, we see them inside the cells, both in the cytosol and in the nucleus. So this shows us that the nanoparticles are able to carry this DNA into the nucleus, where it will then turn into mRNA. So after seeing that these can enter plant cells, we looked if they could actually express their cargo, which was GFP. So in this case, we're looking at expression in the arugula protoplasts. So on the top, again, there's the free DNA. Without nanoparticles, it's not able to enter the cells by itself. But when we express it with carbon nanotubes using the 35S GFP construct, we see it in the cytosol and in the nucleus. And we have also expressed a GFP that was targeted to nucleus. So in this case, we see GFP only in the nucleus of these cells. So after seeing expression in the protoplast, we then looked at expression in the leaves. So here we are looking at leaves after 3 days of infiltration with our particles. And this first one is the tobacco plant again, Nicotiana benthamiana. We have also done this in arugula, wheat, and cotton. And in all of these cases, we were able to see GFP expression in these plant species. So what we saw is that this GFP expression is transient. So the high levels of GFP we observe using confocal at day 3 completely disappears at day 10. And this is at the protein level. We could also look at the mRNA levels of this GFP protein, and we see that it's been stopped making at day 10. So by day 10, there are no more GFP mRNA. So from this, we hypothesized that the proteins could be expressing transiently from the vector, and the vector does not get integrated into the plant genome. So just to show this with a schematic, here when we deliver the plasmid with carbon nanotubes, it enters the plant cell and it makes the protein from the plasmid itself, but this plasmid does not get inserted into the plant genomic DNA. And over time, in 10 days, when the cells divide and this plasmid DNA degrades, we don't have any more plasmid DNA in those cells. In this case, we don't get any more of the protein expression. So this is different in Agrobacteria. When we deliver plasmid or tDNA in this case into the plant cell using Agrobacteria, the protein is made from the tDNA itself, but then also that tDNA gets inserted into the plant genomic DNA. So even after the plasmid DNA is degraded and diluted in the cells, we still have GFP expression from the genomic DNA insert. We showed this also with the qPCR in Agrobacteria, that the mRNA levels will keep stable over a long duration of time as it's expressed from the plant genomic DNA. So to validate this hypothesis even further, we used a method called Droplet Digital Pcr. So this is a very sensitive method that can detect even one integration event out of 500,000 integration events. So when we treat our plants with the carbon nanotubes and Agrobacteria, and after a certain time, when we look at the genomic DNA, we could see if there are any integration. So in these plots, for example, in the Agrobacteria, we are looking at all the genomic DNA we collected from one leaf, and each green dot is an integration event. So in a full leaf, we have a lot of integration events in Agrobacteria delivered sample. But when we deliver the same DNA with carbon nanotubes, there isn't any integration event. So this validated that the DNA, when we deliver it with nanoparticles, is not available for integration, and it only expresses transiently, which lasts for 7 to 10 days. We wanted to make sure that this tool will be applicable in many different species. So after our initial experiments, we have also tested carbon nanotubes in various different dicots and monocots. Here I'm showing some results in Arugula, Arabidopsis, wheat, and some hard to transform species like the dicot cotton and the monocot sorghum, which was very exciting to see. All of these are leaf samples. We have also shown that we can get expression in callus, stem, embryo, and pollen samples. So it also works with different types of tissues, which will be important to do heritable edits going forward. Also, I want to mention that at the concentrations we use them, they are not toxic to the plants. So here we are looking at a leaf that was infiltrated with water, 2 different amounts of carbon nanotubes, and as a positive stress control, we infiltrated SDS. So we could see that the SDS area is having tissue death and necrosis, but the carbon nanotube areas are visibly fine. We could also look at the rates of photosynthesis in these areas. So this is a way of quantifying photosynthetic efficiency. So in the area where we infiltrated the SDS stress inducer, we see low values of photosynthesis efficiency. That means that that area is affected. But in the area where we infiltrated the carbon nanotubes, the plant is able to do photosynthesis just like a non-treated area. In addition to these, we have also looked at some molecular markers of stress in plants. So we picked this gene that's a marker for stress in plants. It's reactive burst oxidase homolog B gene, which is upregulated when plants are under stress conditions. So we could see that in the SDS infiltration, the plants are highly stressed as expected, but in our carbon nanotube treatments, the stress levels are very low, and it's very comparable to just the buffer infiltration. And we have also done some RNA sequencing results that I won't be able to share today given the time restrictions, but we were able to determine this PR1, pathogen response 1 gene, as a marker to look at the carbon nanotube toxicity. And we could see that this toxicity depends on the concentration of the DNA carbon nanotubes we use. At low concentration, for typical experiments, we would use 1 milligrams per liter. The toxicity is low, comparable again with water infiltration. But as we go higher concentrations, its toxicity is also increasing. So that's something to keep in mind when we are using higher amounts. So to summarize this part so far, we were able to develop a delivery tool that is so far species independent, efficient. It is completely abiotic. It doesn't use any virus or bacteria parts, and it does not integrate DNA into the plant genome. We have shown that it has a good efficiency. So it's more efficient compared to a gene gun where we get a larger cell population expressing. But it's less efficient compared to the agrobacteria. So we usually get maybe 30% of what we get when we compare it to agrobacteria expression. So it's somewhere in between the efficiency of a gene gun and an agrobacteria. And as I mentioned, it doesn't cause integration. So this was the first time delivering nanoparticles into plants that didn't use an external force like a gene gun method, and it addressed some of the bottlenecks of DNA delivery to plants. So next, we wanted to use this developed tool for CRISPR gene editing. So all we needed to do is just to replace our GFP plasmid with a plasmid carrying CRISPR parts. So in this case, that would be a Cas9 protein and a guide RNA coded in this vector that we can load onto the carbon nanotubes. And for initial experiments, we designed our guide RNA for gene knockout to cause small insertion and deletion events in DELs and to see if we can use carbon nanotubes for gene editing as well. So we have done all of the gene editing studies in Nicotiana bentamiana in the tobacco plant. So I'm going to show a couple of experiments. The first one we used Cas9 expressing transgenic tobacco. So then we would only need to deliver guide RNA plasmids. So it's an easier place to start again. And we have also done the similar experiments in the wild type tobacco and in the GFP expressing tobacco so that we can knock out the GFP expression. And in these cases, we would need to deliver plasmids coding both the Cas9 and the guide RNA. And in these initial experiments, we targeted the phytoN desaturase PDS gene. So this is a visible gene when it's knocked out that the plants turn white. So it's an easy way to phenotype the edited plants as well. And initially, we used 3 methods to quantify gene editing. So the first one is Sanger sequencing and indel analysis using TIDE. This is, I think, the method that is the, first of all, easiest, less labor, and it's more quantitative and precise compared to other enzymatic-based methods. So the other method that we used is this T7 endonuclease 1 enzyme that recognizes mismatches in the genomic DNA caused by the CRISPR editing, and it will cut the DNA in there. So in the edited samples, you will end up with multiple bands when you run this on the gel. In my experience, it gives a higher background. It's a bit more labor intensive, and you need to be careful designing your guide RNA primers because you don't want to center the guide RNA. The other method that's also enzymatic is the restriction site mutation assay. So in this case, when you're designing your guide RNA, if you design the guide RNA to mutate a restriction enzyme site, so in your mutants, the restriction enzyme site will not be there. So when you extract the genomic DNA and add the restriction enzyme in the wild type, it should be cut into 2 pieces. But in the edited one, because you don't have the restriction enzyme site anymore, you will get a 1 band. So then it's again an enzyme running on the gel type of assay. It restricts highly the types of guide RNAs you can design because it also needs to target the restriction enzyme site in addition to your gene of interest. And it's again, because it's enzymatic, it's a bit more labor intensive. So we used all these 3 methods to quantify our editing efficiencies. So here I'm first showing the editing results of the Cas9 tobacco. So that's the transgenic tobacco that we only needed to express a guide RNA vector. So this guide RNA was designed in this paper earlier where they also knocked out the PDS gene. What we did, we loaded this plasmid onto the carbon nanotubes, and then we did a heat treatment. So the plants were in 22 degrees for 24 hours, and then they were at 37 degrees for 24 hours, and we rotated this cycle 5 times. And we were just trying to increase our editing efficiency, as Cas9 has higher activity at higher temperatures. So I'm first going to just show the results for each of these editing, and then I'll go into the details of quantification a little bit more. So in this case, our editing percentage when we use carbon nanotubes is around 15%, and this is indel generation efficiency. And in the case of Agrobacteria, it's around 40%. So we could see that carbon nanotubes are able to cause gene edits. They are less efficient compared to Agrobacteria in this case. And when we do the similar assay in the wild type tobacco plants, in this case, to knock out the PDS, we expressed a 35S driven Cas9 and a U6 driven guide RNA. It's the same guide RNA from the first assay. And then we did the same heat treatment. This time we see more or less comparable editing efficiencies between the carbon nanotubes and Agrobacteria. So carbon nanotubes are causing 2% editing efficiency, whereas we have around 3%, 2.8% for Agro. So these efficiencies may seem low, but this is just the editing in the leaf. So we are not selecting any edited cells. So when we collect genomic DNA, we get low of wild type that is not transformed, but 2% with carbon nanotubes we think it's a really nice achievement, because just to remind you that in this case with carbon nanotubes, it's a completely transient expression of the Cas9 and guide RNA. So this vector did not get integrated into the plant genome. The Cas9 and guide RNA was only there for 7 to 10 days. They did editing and then they disappeared. Versus in the Agrobacteria, these cells will have the Cas9 integrated. So in the next generation, say this is a heritable transformation, we would likely need to remove the Cas9 and do segregation. So it has advantages of being able to do same or similar editing efficiency without integrating the Cas9 vector. And in the last case, I'm going to show the editing of the Gfp gene. So here we had the Gfp transgenic plant, and we wanted to knock out the Gfp in that plant. And this time we designed 3 guide RNAs targeting different parts of the Gfp gene, just to study if the location in the gene makes a difference. And again, the same heat treatment. So we see varying efficiencies with different guide RNAs from 4 to 8%, but it wasn't a very drastic difference between different guide RNAs. All guide RNAs were designed ideally in terms of their sequence. And what we also did here is that we knew that the carbon nanotubes are not able to travel too far from where we infiltrate them. So they stay typically local around the infiltration area. But when we collect genomic DNA from the entire leaf, that just dilutes the amount of edits we get. So this time we decided to just look at the entire leaf area as well as just locally. And what we see that if we look around the infiltration area, we have 5 times higher editing efficiencies. So again, this is something to keep in mind when we are using these nanoparticles. The editing events will be around where we infiltrate them, and it's not going to go further away from that location. So these are the broad results that we obtained using carbon nanotubes for editing. But I also wanted to show how we actually did the verification and quantification of this using those 3 assays that I mentioned. So I'll just show this for the transgenic Cas9 tobacco plants. So in this case, the first I'm showing is the restriction site mutation assay. And then initially we were doing these without heat treatment. So we are looking at the without heat treatment result here. So this is the amplified PDS gene before we add the restriction enzyme. So it's not cut here. We have the PDS gene sequence. So we have a non-infiltrated sample, 2 Agrobacteria infiltrations, and some negative Cas9 plants that is not infiltrated or infiltrated as agros. These are negative controls because when we don't have Cas9, we should not get any editing in these plants. So then what we do, we treat this PDS gene with the restriction enzyme. So in the case of non-infiltrated sample, so there is no editing in this one. You could see that the band at 450 disappears completely, and you have 2 cut bands. This means that the sample is non-edited, as there aren't any gene copies that are resistant to the restriction. Whereas in the edited samples, we could see that because the editing is not 100%, we still have cut bands, but we also have a restriction enzyme resistant band at the top. This means that this 15% of the cells did have the edit. So this is how we quantified. And in the negative controls where we didn't have Cas9, there was no editing. So this is without the heat treatment. We thought that these 10 and 15% were a little bit low. So then we did the heat treatment and did the similar assay. So what we see here, in the case of Agrobacteria, the editing efficiency was 44%. So almost like half of it was not cut and then half of it was cut. So we could see that the heat treatment really increases the efficiency. And in this case, we included our carbon nanotube samples as well. So we saw that there is a faint band at the top. This corresponds to 13 to 14% editing in the carbon nanotube samples. So this is how we quantified using the restriction site mutation assay. Similarly, we could use the T7E1 assay. So this is the one where we can use this enzyme to cut the edited copies of the genome. So in the case of non-infiltration, we don't have any cut bands. So this means it's not edited. In the case of agro-positive and the carbon nanotube positive control, we have cut bands. But as I was mentioning, there is also a lot of background bands, and it's not always the cleanest way of identifying gene edits. But it gives similar editing efficiency with the restriction site mutation assay when we compare them. And then the last assay that I think is the most quantitative and always gives cleaner results is the Sanger sequencing of these PDS amplicons and running a tight analysis on them. So here we are looking at one of the agrobacteria samples, and it shows us that like 38% of the genome is not edited, but we have total of 35% that is edited, and it also shows the breakdown of these edits. So it tells us that like there is 16% plus 1 insertion events, 7% minus 1 deletion events. It gives us a breakdown of all the different types of edits. And we can similarly look at this for the nanoparticles, where we see a total of 15% editing efficiency. Most of it, 10% of it, is a +1 event. So these are the 3 methods that we use to quantify our gene editing when we delivered CRISPR cargos with nanoparticles. So this was a more or less recent work, and there is a lot more ongoing work that's happening in the Landry lab right now. So just to summarize, we got the highest editing efficiency in the Cas9 transgenic plant. We had lower editing efficiency in the wild type plants, but comparable between agrobacteria and carbon nanotubes. Guide RNA design is important, but with today's softwares, it's easy to get optimal guide RNAs designed. And closer to the infiltration area, we have higher editing efficiencies. So what's happening right now is optimization of the carbon nanotube amount and infiltration conditions. We have been trying the Gemini virus replicon vectors to increase editing efficiency. We are improving our constructs by optimizing for codons and promoters. And lastly, we want to look at the off-target effects using a whole genome sequencing. And we are trying to translate all of these editing we have done on leaves into heritable transformations using developmental regulators. So as I mentioned, we have interest in being able to do these edits heritably. Maybe flowers and pollens and shoot apical meristems could be nice tissues for this. In addition, I think it makes sense to use carbon nanotubes for transient expression of morphogenic regulators and developmental regulators. So these, when they are expressed transiently, together with the CRISPR cargos, will give rise to gene-edited new embryos. So it could be a fast way and maybe tissue culture-free way of generating edited plants using the nanoparticles for transient expression of these tools. And more on the chemistry side of the things, so far we have only developed methods for the plasmid delivery, but we also have interest in delivering Cas9 as a protein and a ribonucleoprotein complex as well. So we are trying to develop some chemistries for these different cargoes. So all of this work was done in the Landry Lab at UC Berkeley. I have a lot of people to thank there and a lot of funding sources for this work. And I did have a Schlumberger Foundation for my PhD work as well. So I would like to acknowledge all of these people and funding sources. So in the remaining 15 minutes I have, I'm going to talk about my postdoc work that I'm currently completing. I'm doing this in Professor Shubham Brady's lab at UC Davis in Plant Biology Department, and we are hoping to engineer nutrient use efficiency of tomato plants, again, through CRISPR gene and epigenome editing. I'll start by saying why I'm interested in studying plant roots and nutrients. And that's because we and the plants are facing changing environmental and climate conditions with increasing temperatures, excessive availability of water or drought, and a lot of new emerging pests and diseases. All of these decrease the crop productivity and yield. Given these changes in the environment, as well as the increasing human population, we need to increase the crop productivity, but we need to be able to do it in a sustainable way. So over the last century or so, we've been using a lot of chemical fertilizers in an unsustainable way just to increase the crop yields, but these fertilizers had negative consequences on the environment. So if we could understand how plant roots respond to the environmental conditions, the availability of nutrients, so that we could engineer these root systems more efficiently to be better adapted to the changing conditions, as well as to support the growth of more productive plants using less nutrient fertilizers, that would be a very impactful way of going forward with some of our gene editing studies. So in my postdoc lab, I focused on nutrients, and I specifically worked with nitrates and phosphates. These are essential nutrients for plant growth, but their excess usage has been causing a lot of ecological and environmental consequences. So we need to understand how plants use these nutrients so that we can do precise edits to use as minimal fertilizers as possible, but still have higher crop yields. So this is what I've focused during my PhD. I'm going to try to not go into so much detail here, but I'm initially focusing on the nitrogen metabolism regulation. And I know this looks very complex, but what I'm showing is actually, this is in Arabidopsis. This is a gene regulatory network in the center showing transcription factors regulating a lot of different genes. And this is only for the nitrogen. So all I'm trying to show here is that this is a very complex network of genes and proteins in Arabidopsis. And as we all know, Arabidopsis is one of the smallest genome plants with only 135 million base pairs. And when we translate to tomato and many other crops, we have huge genomes. For example, in the case of tomato, we have 950 million base pairs. So you could only imagine that how this nitrogen network is going to look in tomato and how hard it is to understand these interactions, yet alone edit them. So this project focuses on trying to understand some of the key transcription factors in the nitrogen network. And it's a collaborative project between Brady Lab and Nicola Patron's lab at UK, where we are looking at these transcription factors and how they regulate nitrogen metabolism, both in Arabidopsis as well as in tomato. So our aims is to look how much of this pathway is conserved between these two species, because most of the research is done in Arabidopsis that aims to increase the nutrient use efficiency, but we need to understand what we can translate in between Arabidopsis and different crops. So how we do this, so our aim was to create the single and higher order mutants of all of these transcription factors in tomato. And because transformations take a long time in tomato, we use a different system. that is based on hairy roots. So if you're not familiar in hairy roots, that's a fast way of doing CRISPR transgenics or other transgenics in crops like tomato. So we start with cotyledon, a piece of leaf, and we transform it with a bacteria called rhizogenes, Rhizobium rhizogenes. So this bacteria carries a Cas9 and guide RNA, as well as a kanamycin resistance. And after a couple of weeks, we started to get these roots called hairy roots from the transformed leaf pieces. So each of these roots will have a different genotype. These are all independent transformation events that we select on a kanamycin plate, and then we can propagate them on plates like this. So this is how it looks. You start with leaves, and then they start giving these root pieces. You can transfer an individual root onto a plate and culture it like this. So this is a mutant of like one of the transcription factors, for example. And we can genotype them, and we see a lot of different types of CRISPR edits, large deletions in some cases, or indels. In tomato, we typically use 2 to 3 guide RNA per gene to make sure we get nice edits. So in this case, we did all the single and combinations of mutants in this network. So you can see we have 7 transcription factors. So we had constructs that contained 21 guide RNAs in some cases. And how we do such complex editing and cloning is using this multiplex mutagenesis strategy that was developed by Pollack et al. So this is called loop assembly. So it's a way of cloning very large vectors with a lot of different parts. If you look at it, it has the region where you have the guide RNAs, Cas9, resistance, and some selection as well. So technically, it gives you a way of doing a limitless amount of insertions. So as I was saying, we have all 7 transcription factors mutated. So that means 21 guide RNAs in this one vector that we were able to clone in a very easy and fast way. It has a marker kanamycin for the transgenic plant and root selection, and it also has a seed marker. So this is fast red. It turns the edited seeds into red, so we can easily pick the ones that are edited with a dissecting microscope. So what we are doing currently with all these lines, because we have so many lines, we are genotyping them by deep sequencing of PCR amplicons using a facility at Harvard Medical School. So they are giving us the types of edit and the percentage of edits in all cells we have. All lines we have. We are phenotyping these mutants under different nitrogen conditions, and we have already identified some promising lines where the knockouts gave us a higher nitrogen use efficiency and nitrate responsiveness. So we were able to see some of those transcription factors and interactions being able to grow these tomato with less nitrogen input. So because these hairy roots are not real stable transgenics, we are in the process of creating stable mutants of these promising lines through callus-induced mutagenesis, traditional way of doing the transformations in tomato. Okay, so with that, I would like to also thank my postdoc lab, Shubham Brady, and all of our collaborators at Berkeley, Israel, and UK, especially Nicola Patron's lab. And thank you all for listening. I'm happy to take questions now.
Okay, Gözde, thank you very much for this amazing talk. So right now we can take a few questions from the physical participants first, then we will go to the online ones. Yes, please. Can you come to the stage to ask the questions? Thank you.
Hi, Gözde. I am Nihal. It was a very nice talk. I'm Nihal from Sabancı University Nanotechnology Research and Application Center, and we want to replicate your research about carbon nanotube and the delivery of the vector to several actually crops. My question is a bit technical. I want to ask you what other ligands that you can use instead of PEI to attach plasmid onto carbon nanotubes? And I have another question after that.
Okay. Yeah, great question. Thank you. Yeah, I didn't go into the details of the chemistry today, but PEI was the positively charged polymer we used to be able to condense the DNA on the carbon nanotubes. And we are trying to switch it with different types of positively charged molecules because the toxicity I showed in the RNA sequencing data, that is caused by the PEI. So when we use high concentrations of PEI, we end up getting a lot of toxicity. It's more manageable in the leaves, but it became a problem when we were working with the protoplasts and tissues that we tried to regenerate. So we are trying a huge library of positively charged polymers. I think one of the one that worked as good as PEI is polylysine. So that's an alternative that I would suggest. It's a bit more expensive than PEI, but it has a lower toxicity, significantly lower toxicity. So that's an option. I'm not sure of any other one that has shown success yet, but I could check it out and let you know. So this work has carried on after I graduated, so I know that they've been looking at different molecules as well.
Okay, thank you. And I have
sorry, there's another one actually. There is chitosan, I think, as well, that they had success loading DNA.
Okay, and also, can you tell me the maximum size of the plasmid that you can use for the delivery?
Yeah, that's also a great question. So it's more efficient with smaller plasmid. So the initial optimization we have done, they're all with 4 to 5 kb plasmids, which is not realistic really with when you're looking at CRISPR plasmids with selection markers. We did get expression from things up to 12 to 14 kb with a lowered efficiency. I have not tried anything larger than that.
Okay. Thank you so much.
We can take one more question over here. Sadi hocam, please.
Hi guys, a great talk. Thanks for your talk. And my question about, you told you transfer them like infiltration, but have you tried in wheat leaves? But have you ever think about the regenerate plants from the leaves, like the?
Yeah, we haven't tried that. Yeah.
That a morphological gene technology that we can use them for the increase the regeneration and for the somatic embryogenesis, and we can regenerate plants from the leaves.
Yeah, exactly.
So
that's
kind of study.
Yeah, I think that'll be a great way to go forward. We haven't tried that. I think it'll be easier than like trying to regenerate from callus. I think because we know we can do the leaves, so then we could try to regenerate from leaves. What I've been suggesting to do for future is to transform the shoot apical meristems, especially in tomato, they are pretty visible. So you can inject the carbon nanotubes together with the developmental regulators into the shoot apical meristems in the leaf axillary meristems, and then hopefully you'll get new branches that are edited that you can collect seeds in the future. So that's what I've been trying to develop in tomato right now with the shoot apical meristems, but I haven't tried the leaf regeneration.
Okay, because you thought you can transfer them to infiltrate with leaves mostly. I see your presentation, but you can use ITEX in immature embryos as well, right?
We haven't tried embryos yet. So I think you may need to optimize for embryos, like the concentrations and the protocol, but you could just, you can put it in a syringe and inject into the stems or the meristems, for example.
You can regenerate transgenic plants from the leaf explants, instead of leaf explants. It would be interesting to try in meat like this, I thought.
Yeah, I think so too. I think it'll be really exciting to try.
Okay, thank you.
Thank you.
Now we will take one more question.
So, one part of my question is already asked by Haita, so I'll ask the second part. So there are reports that if you dip roots in carbon nanotube, probably they can take up the DNA and that goes into the upper parts and look maybe very low efficiency, but if it works, then it works. Your comment, please.
We have not tried that either. I did see those papers, yeah, through the roots, and I will. and then it increases the germination rates. So I think there are some benefits of having carbon nanotubes in the roots and in the germination media, but we haven't tried delivery through the roots. So yeah, I don't have much to say on that because we haven't tried it.
Okay, we have one more question from the physical audience.
Hi, Jazda. Thank you very much for your very interesting presentation. Actually, I am not expert on the nanotechnology, but I know that in the medical system, there is nanoparticle-based tissue-specific delivery system. Is there any tissue or cell type specific gene delivery system in your lab or in other part?
Yeah, I think that'll be really nice to develop. I think that's one of the advantages of the nanoparticles, that we can take advantage and use in plants as well. So I think the easiest would be to target by pH, because we can have nanoparticles that are responsive to pH. So then like the cell wall pH is lower, vacuole pH is lower. We could potentially target those compartments. And we can also take advantage of targeting peptides on the nanoparticles, because we know sequences of targeting peptides for the chloroplast and mitochondria. So we could attach those peptides on the carbon nanotube surface to put them into the organelles, but that's something we haven't tried yet. I think we did try to attach some peptides on them, but the stability was an issue. So the protocol, we need to optimize it a little bit more. When we have like both the DNA and the targeting peptides, the complex was not stable enough, but I think there's a potential in investigating those.
Thank you very much.
Okay, now I will also look to the chat box to see if there are any questions from the online participants. Let me see that. Okay, it seems that there are no other questions there. Dear Gözde, thank you very much for joining us. I know it is too late already there. Have a good night, and we appreciate your presence here.
Yeah, thank you so much, and thank you all for great questions. Have a good rest of your day.
Thank you, bye. Okay, dear participants, we will continue with our next speaker, dear Dr. Sadiye Hayta. She's together with Mark. They are from John Innes Center, and Dr. Sadiye has been working on wheat transformation, and she has developed an interesting, very important and very efficient protocol for the transformation of the wheat. And I think today she will talk about this transformation efficient protocol as a case study. Thank you very much for joining us. Let me just şunu bir açayım ilk önce. Ondan sonra da buradan ne oldu? Buraya nasıl geri geçeceğiz ki acaba? Bunu tekrar nasıl açacağız Zoom'u? Orada olduğumuz için. Pardon. Evet, sonra. Bir de şununla mı açılıyor? Lazer.
Thank you, Emre, for introduction, and good morning, everybody. I'm Shadi Haite and I work at Journalist Center. And today I'm going to talk about our transformation system. I work at Journalist Center, and Journalist Center is a part of Norwich Research Park. And I'm also work in the crop transformation group and responsible for wheat transformation and genome editing. In wheat, we've done so far gene overexpression studies, gene complementation, RNA interference, and mostly genome editing. Actually, yesterday, Mark explained wheat is one of the most difficult crop, and it has got very big genome. And with traditional breeding and mutation breeding, it's very difficult to use in wheat because wheat has got big three subgenome. If you just mutate it, if you cast out one mutation in one subgenome, can you hear me, or shall I just stop? Okay. Can I use this mic? Okay. Traditional breeding in wheat very efficiently. Second method, you know, as everybody knows, is mutation breeding. But again, you need to knock out three mutations because which really we don't want. Polyploid plants like wheat is the crisp before. And if it is or maize, if you knock out one genome, and you can see that this kind of 21% increase in grain weight. But in wheat, you need the knockout 3, you can say 6%. If you do double mutation, 11%. And if you knock out the 3 subgenome, you can see the 20% grain weight. Is a problem with, okay. Our strategy in wheat is depend of our target because we are working in wheat researchers at Journalist Center, also not only Journalist Center, other wheat researchers as well. Usually they send our target to us and we just check them. We can use, depend on their target, base editing, multiplex editing, even the prime editing, but mostly of course knockout. And we first, yesterday Mark explained all of them, but first we design our guides. And usually we target the first incron, and mostly we use 2 guide RNAs.
I think the Zoom people cannot see us. Let me just...
Again, I was talking about our strategy in genome editing in wheat. And usually when we design guides, we use the 2 guide system, but it depends on, again, our targets. And we can use now 6 guides in the wheat transfer with genome editing. And for each target, we use 2 construct, and each construct got the 2 guides usually. And then after we prepare our construct, we transform into wheat. In wheat genome editing, actually also all the genome editing is one of the important factor is the good sequence data and tools for designing guides. If you haven't got any good detailed sequence data, you can't design it. And in wheat, again, big genome and the good sequence data really revealed is recent in 2018. I will tell you a bit later more details. And second important factor is an efficient delivery system. It can be biolistic or Agrobacterium, we use the Agrobacterium transformation protocol. And also the screening is also the important factor, especially like the wheat, polyploid wheat plant, it's more challenging and you need to produce a lot of plants to screen it. In wheat, the good high quality genome is just released 2018 and it was in Chinese sequence. And years we use the Chinese sequence as a genome sequence for design guides. And we transform fielder. But recently, especially in the last 8 years, with a lot of high quality hexaploid or tetraploid with genome release, and we can now use it efficiently in our CRISPR work. Can I use this one now? Okay. As I previously say, I work with the with researchers and they examine of their genes, and we work with them to assess their gene function. And after that, we see that if we are working in Kronos or Paragon with each variety they send, we design the guides for them and transfer them to in vitro. Second factor is efficient delivery system, which considered one of the most difficult crop to transform, and it is very genotype dependent. The first transgenic plant is obtained in early 90s, while Agrobacterium mediated transformation remained around 5% after that. And there is a remarkable protocol in 2015. It is a patented licensed protocol, and it requires special training and vector, so it can't be used very efficiently. Our open access protocol is published 2019. It is an efficient protocol, but still it is genotype dependent. And 2020, with Juan Dabrowski-Rapp in UC Davis, we published a protocol. It's increased the efficiencies in with transformation. Also, it's increased regeneration, transformation efficiencies, also reduce the genetic dependence drastically. In our transformation system, we use Agrobacterium transformation system, and we use different wheat variety fielder. And our efficiencies is good for the fielder's efficiency. It was 33%, and the Kronos we can get 10%, in Cadenza 5%. It's great for the wheat varieties. But our transformation system, like not ours, the others as well, affected from a lot of factors, and wheat is more sensitive than other crops. Even the growing conditions, small change, or growth donor plants had a big change, these factors are dropped drastically. We use Golden Gate vector system for our transformation protocol. It allows us to clone quickly and efficiency. Also with the combination of PGG vector with our helper plasmid, which contains a VRG, give us higher transformation efficiencies. We use gas reporter construct for initial our experiments for optimization of transformation protocol. And if you got interested in our transformation system and constructs, you can contact the Mark, or you can find our constructs all in HGene. In our transformation protocol, we use immature embryos, and this is the stage the transformation affect a lot. If our plant material is not healthy or in wrong stage, or transformation efficiency is affected drastically. We have a centrifugation stage before co-cultivation. Leaf samples for DNA extraction, and we check the edits. And I just want to give us some of examples from our transformation studies. In this study, we Thank you. A second example I would like to share with you today is our collaboration with Brand Wolf. He's now in CAST, and he's actually, if you're interested, he's looking for postdocs and PhDs. And this is, it is in stem rust resistance, and we just check the resistance transgenic plants and compared with what type, you can see that they are sensitive and they are more resistant. And it's patented now. Now we start to use a protein fusion growth regulating factor, and is it a cofactor GRFT in our transformation efficiency, and now we can, and also it shortened our transformers is not affected from other external factors. My first results from the paper we published drastically increase, and they don't affect any other external factors. Now our transformations, running our transformations efficiency around 77% in filter and 70% in Kronos, especially Kronos gain more benefit from this system. We can transform mature embryos. I just tried, so we don't need to grow plant matters in in CRs or glass house. So we just pick up from shelf and we can transform them, and efficiency. It was my first try, and efficiencies were in 9%. Also now we know that we can transform cyclic yeast as well. That's why I asked Gözde this question. And this is our control place, and you can see at week 7 with the transform plant with GRF. The GRS technology and plus our transformation system allow us to extend the transformable genotypes more. Now we can transform Paragon and Cadenza, which we have got really good genomic resources with. Also, we know we can transform some elite commercial wheat varieties like Sky4 and Redlink. They are the recommended list in UK. And our efficiencies again is very good. You can see the Redlink, you can transfer 40% now. Previously, we thought we used the model plants for wheat, but in last meeting, we just thought we start to get a model plant now. We get a real genomic resources. We can use Paragon, Kronos, and Cadenza genomic resources directly, and we can transform the directly plant. So we don't need to use the model plant now. Unlike the other morphologic genes like the baby boom and visual system, we haven't got any detrimental effect in our plants, especially in low copy numbers. But we noticed that if the copy number is higher, GRF expression higher, there is some undesired phenotypes which can confuse with our gene function. And some of its varieties are more sensitive than the others, like Kronos. So we start to develop other systems, we can control our GRF system. Now Mark working on it and prepare some construct for us, and we start to test them. First one, we use is Krylog system. With Krylog combination with the heat shock promoter, when we increase the temperature, we are just in later stage, we can, it allow us to remove GRF in later stage in the transgenic plants. The other one is the controlling the GRF. We use the PLTP. It is the endosperm embryo-specific promoter, and it doesn't express in later stage. So we can produce plants, we can produce callus, somatic embryogenesis, but and later stage, it doesn't express the GRF. The other third system Mark prepared is the transformation. We aim to use 2 separate tDNAs. It means it's going to allow us to segregate away in later stage, and also we can use the previous construct. We just use it for filter, so it doesn't contain any GRF. So we can just use them together now, so we don't need to prepare new constructs. Third factor, I think one of the most important, also time-consuming, was screening, especially for polyploid species, because usually we just PCR them, amplify the gene, and after that send them the single sequencing. But like with each individual subgenome needs to be screened for EDIs. This requires at least 3 PCR reactions and 3 sequence reactions per plant. It is a bit time consuming and you need to optimize your PCR. So we start to use now next generation sequencing, especially if we are targeting to 3 subgenome. This was our first published method, and it was targeted at only 5p copy in wheat. But it is actually a bit challenging because targeting, if we had a choice of 3 guys only, so it takes our time. So, but we were able to obtain a big plants with the large deletions, which contains 114 base pairs deletion. Because the deletion was large enough, we just screened them with PCR and you can see the large deletions. This is homozygous EDIS plants and these are heterozygous plants. And this is the wild type genotype. When we sent the Sanger sequencing, because it is just in one subgenome, you can see the large deletion by sequencing. Also, it is with the Cristobal and Cristobal's team, we tried, in this target, we just targeted a genome. So we just directly sent them, we PCR'd and sent them the Sanger sequencing again. This is again Cristobal and Cristobal's PhD students target, and in this one they were targeted promoter region and they used the 3 guys, and they didn't check any SNPs and small deletions. Just their target was large deletions. When that's why they only PCR'd, and you can see they got the 6% in Fielder and 7% in Kronos. They get some large deletions, and they even sent the Sanger sequence for it. And when we start to check the next generation T1s, it is most interesting because it shows us the deletions, actually mutations carry on the next generation with CRISPR. The plants haven't got any deletions or mutations in first generation, they got deletions in T1. As you can see, it is 19% and Kronos is 9%, or almost around 10%. So it was very interesting for me to see the next generation, you can get more deletions. This is again with our work with Graham. He is the design feature with strategic leader. And this is our work with the collaboration with the Danforth Center in the United States. In this target, we were targeting the 3 subgenomes. And you can see that we got homozygous EDIs in the 3 subgenomes successfully. The previous sample was in Fielder, and after that we tried something in Kronos, and you can see again homozygous EDIs in the 2 subgenome successfully again. Now, we are working on another system as well, because we always use the transformation system and tissue culture. But this method, high edit protocol, is allow us to produce edited lines. We may upload in edit with target genes. And maize, you can see that it fertilized after maize chromosome elimination, we can get homozygous edit without any transgene in it. This is our most recent detailed information and CRISPR system. Both our papers are open access and complement to each other, and we have videos with them. Also, versions of other transformation method, you can see the bit training, it's arranged by Cristobal's and Cristobal's team. And all the vectors we use, you can find in Gene. I would like to thank the people we work together. I would like to thank the crop transformation and genome editing platform, but especially my team and Mark for the bit transformation. We are the new team, but Our results are very good now, very good at the moment. Also, I would like to thank Christabas and Graham's group, and also UC Davis, our collaborators. They are Juan and Jorge. They are amazing people, and they are open to work, and all the researchers at John Innes Center. Thank you.
Thank you very much for this great talk. Now, we can take some questions from the audience, please. If you can talk right in front of the can you come over here so people can see you?
Okay. First of all, thank you for such an interesting presentation, and also Dr. Mark's presentation was so interesting for me. You say with sensitive than other crops. Why you think that? It's my first question. And I am a physiologist, and it's interesting for me. Equal to these changes at the molecular or genomic stages in the wheat. Did you investigate biochemical or physiological changes in the wheat plants?
I answered, I just tried to explain. Okay. I think we can say a recursive sequence. That is why it is the tissue culture response is very slow and very difficult. Only both white and fielder have some good tissue culture response. That's why maybe first transformation start with varieties. What we always consider one of the most difficult crop to transform, and it was really because when I started, it takes our time to optimize all the protocol. But the GRF system make really revolutionary change in the wheat transformation for us. And usually we are working and collaborate with other people, but physiologic, some different targets they offer us to work on it. But can you repeat the second question, please?
Did you investigate physiological or biochemical changes at the your wheat
Transformation?
Yes, transformation plants.
Actually, we don't check. We just looking all the full parameters, how our transformation affect the phenotype and other factors, but we haven't checked the phenotype stuff. Mark, have you got any? Because usually people working about like iron or stat, and they just approach us, they got the target gene, and we just blast find the sequence, and we design our guys or think about which strategy we can use for that. So we produce transgenic brands, check the copy number analysis for them, so they can compare their results, and they give DNA sample and plans them to carry on their research. So each again depend on our target and which people can approach us. If they ask us to carry on their more research, we can carry on it for them.
We can take one more question. So, can you come here because the audience online they cannot hear this.
Yes, I understand. Thank you very much, Dr. Sadi Haider, for wonderful talk, and I'm very excited. So, can you give a kind of a timeline from the design of guide RNA up to the first generation plant? How much time it is taking now?
It takes 11 weeks from beginning to finish the transformation, but it's just only tissue culture stage until we transform them to soil. But with GRF technology, it's getting shorter now. But we can say the design will guys, because a lot of them has to be transformation. So we need to just prioritize the works. But usually it takes 1 year. We design the guys, put in the wheat and transfer them soil, DNA extraction, copy number, and start to check the edits. We can check the edits for other people, or people if they want, they can put this stuff.
So
but
probably
the more challenging part is screening now at the moment. But
Yeah, but I think that, probably maybe
We can say 1 year.
1 year or even less.
It won't be as fast because now some people just approach us and give out the target and they ask us, our construct is ready, and we say, yes, your plans are ready now. Because with the GRF system, its transformation procedure is very shortened and we don't need to repeat anything.
This is amazing progress. I think that, let's congratulate John and his team, and we hope that, we'll be working together with all this. We were able to, at our institute, we were able to produce, transform all the varieties, but your efficiency is really good. Thank you.
Thank you.
We have one more question. Allah.
Thank you so much. Nice presentation. Wheat is a polyploidy crop. How much challenging it is to generate genome-edited plants from such a high polyploidy crop? And for example, if you want to generate a knockout and getting a knockout with 6 alleles knocked out, it's challenging. How much challenging you find?
No, I think it's getting easier and it's for us a routine because we are doing a lot of demand CRISPR. CRISPR is now, I don't know, everybody very popular and everybody interested in CRISPR. So we are doing CRISPR work a lot at Gmo Center and it is our routine work. So most of them we just targeted 3 subgenome because you can, otherwise it's mask and you can see the phenotype. For us, it's routine process. If we can see the T0, mostly we can catch T1 homozygous edit in 3 subgenomes.
Do you often get a kind of a situation where some of the alleles of the same genes are edited well, others not? Because of some allelic diversity and all those things?
When I checked them, the next generation screening, it was in T1s I checked. You can see the A, B, and A, B, D, or D. So it is nice to comparison, especially it's reported for gene.
All right. Okay, thank you.
Do we have any other questions from the physical participants? Otherwise, let me check the chat box. Okay, it seems that we don't have any questions from the online audience. Thank you very much, Dr. Sadi, for joining us. Thank you. Thank you. We appreciate it. Now we will give 15 minutes of break, and right after that we will join again one more time.
Incoming, I think they should submit it on the desk.
Okay, now for the online participants, there is a poll. Could you be able to fill it and submit it in the next few minutes? Thank you so much.
Okay. Ladies and gentlemen, thank you very much for this session, and it's a pleasure that we have Dr. Ismail Abdul Hamid. He is from IOFS, and several people asked that what can be done and what are the how we can keep this group going in the future. So I've especially requested Dr. Ismail to give his comment, and I'm sure that with his support and the support of all our colleagues, there's a lot of enthusiasm and a lot of progress made in the last 2.5 years when we met in Islamabad in 2019. And congratulations to Dr. Emre for arranging this meeting. With these words, I request Dr. Ismail to speak. And especially we think that by having some common understanding and coming up a program, especially on wheat, will be a great service to the whole humanity and also OIC member state. Thank you.
Thank you. Good morning. Thank you very much for everyone that gave me the thank you. Well, I'm actually we are running 16 programs, and they are all tackling the food security. And we are interested almost to cooperate with our member states. OIC has 57 member countries. 36 of them are already members of IFS. So we are not only working with 36 member countries, but all OIC member countries. We work in strategic commodities, which is wheat, rice, cassava, and palm oil. And we are interested also for management water in agriculture. And how can we conserve our genetic resources? I think this is most likely in science. Among the other 16 programs, it is in IFS. We are not actually funding agent more, we are cooperating with each other. We work for our member countries, and if we work for wheat, for instance, we could not work alone. We are not a specialist in wheat, but we have to invite a high caliber from our member countries or even outside member countries in collaboration ideas to write what we are today. Actually, we are not involved today. It is COMCEC-COMSTECH and Middle East Technological University. That it is, I came today also, thanks for the invitation, for inviting me to come, and also to start the channel and the bridges of linkage with the eminent scientists in this field. With COMSTECH, we will do a lot of activities during the 2022 and about action plan. And we are all talking about science. And we also cooperate with SMIC for halal food and halal and food safety. And we are cooperating with CISRIC. It's also for databases and food balance databases. So we can, our activities, we are thinking now to continue our activities. We have a new vision. The new vision will not go to concentrate only on those core commodities, but also we will make it a wider vision, because we have called from other member countries very important about date palm, about olive, and about potato, and about maize. IFS could not work in all of this alone, but we can work and coordinate with universities, with research institutes, with other OIC relevant institutions that we can make this possible. So we are welcome that if you can, like the meeting today, or the training workshop, this one, we can make it next year in collaboration, for instance, with collaboration with COMCEC and the university in another member countries, maybe in Pakistan, maybe in Kazakhstan. So we have to make this kind of activities. You can also at any time contact me, because we always need high caliber of scientists that to participate with us and to make our activities possible. We in IFS need you more than you need us. We are always tackling, and please also you can send me an email at any time. We publish a newsletter about food security hub. We send it to the ministries of agriculture. We send it to head of universities in 57 member countries. So it will be very important to give promotion of your science or what you are doing for. It's not scientific review with journal. It is a bulletin that we tell the people what we are in our member countries standing for the food security. And I think if you talk about food, wheat and rice, it will be very important and high value. So please, if you have any comment, any question, I am here for the next 1 hour, and I'm here now because I do not want to take more of your valuable time, I know. So
it's a pleasure.
Thank you very much. Thank you.
Thank you very much, Dr. Ismail. So his contact will be available to all participants, and Dr. Andre will share with everybody. Thank you very much for your time, and I think that I see amazing progress in the last 2.5 years from the time we met in Islamabad. So very pleased to see all this happening. Thank you.
Okay, thank you very much. Now we have the next speaker. In our program, our next speaker, Dr. Allah Baksh, is coming from CMB in the Center of Excellence in Molecular Biology, right? He has been working on transformation, construct development, and recently genome editing in plants. I know him that he was working in potato transformation for the last 6-7 years. And before that, he worked on many other crop species. So today, I would like to invite him to give a talk. This will be an example to all these previous talks, basically on the development of genome-edited potato lines. Thank you very much, Doctor.
Thank you for the introduction, but this may not be the example for others. Okay.
Now we need to
Online participant can see.
Now they will see 1 second. They will see the share it. We will make it full screen. Okay.
So yeah, laser.
I don't think.
Is there laser?
It's frozen.
Yeah.
Pointer C.
Thank you, Emre, for nice introduction. I put first slide for those whom I haven't met during this event. So basically, we come from University of Punjab. It's located right in Lahore, in the center. So the university was established back in 1882, when British were ruling this part of the world. So my Center of Excellence, is a part of this university and funded by Higher Education Commission Islamabad. And my director, the next speaker, he will introduce with you that center. The center has different sections, and that deals with forensic sciences, stem cell research, some bioinformatics groups, therapeutic groups. So I belong to plant molecular biology. Plant molecular biology is one of a bigger group of the center working on different areas. If I say that mainly what we do, we try to introduce traits of economic importance in croplands. Cotton is the main crop, and among the traits, insect and herbicide resistance is the one that the cotton that already been commercialized, and it's in the country for general cultivation for last couple of years. So this is what I can say that the center has done extremely good as far as the applied side of molecular biology is concerned. So besides that, projects have been conducted, are going on virus resistance. One group is working on improvement of seeds in different crops. And one group, we produce different events of as a result of transgenics. And one group is working on risk assessment studies of all those transgenic crops. And the next speaker, director, he's in charge of that group. So one group is working on edible vaccines for poultry. Basically, one of the group, their purpose is to improve the physiological traits in plants, especially in cotton. And why I'm saying cotton? Cotton is the major crop and the soul of textile industry in Pakistan. So we are also working for the fiber improvement of this crop. And to address all these challenges, we are using molecular tools like everyone. And recently, previously we were using all these overexpression approaches, then the RNAi, and recently the genome editing techniques. And among genome editing techniques, the talk of the town is CRISPR-Cas, and we started working with that. But here in today's presentation, I'll present some of my work in Turkey, because I was working in a Turkish university in Central Anatolia for the last 7 years. And some of the research I will present from this center that was not conducted under my supervision, of course, but my colleagues. But the two examples will be from our Turkish group, and that may not be a complete examples, kind of the research work that is still going on. Potatoes, the importance of potato is just like crystal clear. No need to explain about its importance. It's a hidden treasure, and we say that because we were in central Anatolia of Turkey, that is the best for potato production. And there we started working, and likewise, potato is also very strategically important for Pakistan as well, and of course for the world. And whenever we talk about the production, Turkey and Pakistan, they're almost at the same page as far as the production is concerned. And how important the potato crop is for the food security. And also when we talk about the climate change, potato again has a very important role in that.
And as far as whenever we talk about the nutritional security, potato is one of the crop that can address the challenges. And this is what the journal is believed. And this is the report that can tell what kind of nutrients you can find. Because we were working in potato, so we developed transformation system. Like in a lecture before me, Dr. Sadia, she was talking about wheat, so one of the monocot. When we talk about wheat and potato transformation, definitely potato is a little bit friendly compared to wheat. So, we developed a transformation system using a reporter gene, the GUS gene, that was interrupted by an intronic sequence, and we transformed with Agrobacterium, and we use a combination of different plant growth regulators, and we were able to establish a good transformation protocol in to make potato. And yeah, but using this protocol, we were able to conduct a couple of projects of TÜBİTAK in Turkey, and the same protocol is going on in my current institute. So this is one of the example in our current institute. Anila, she is a PhD student, also the research officer there. And she is trying to knock down this vehicular invertase gene. And the function of this gene is well established in Scrooge's partitioning. And also we percent of the edited region was found as so I'll keep on moving. Yesterday, there was a question, how do you select your vectors? And this is the beauty of recombinant DNA technology. You can design your own vectors, and there is a lot of talk of the town about the choice of promoters, choice of terminators, the people's public concerns about the GMOs. Keeping in view all those concerns, you can make your life easy by selecting different promoters, different terminators with your gene of interest. And also there is how we should drive Cas9 with different promoters. One of my students he conducted this work, and Ilham Raham Kolov, right now he's a PhD student. This was his master work. And we just tried to drive this Cas9 with different promoters. And the promoters, what was the workflow that was, first, again, the concern, among concerns, one of the concerns is about plant selectable marker. And among plant selectable marker, we try to remove hygromycin, replace hygromycin with the kanamycin as a first step. Then we change the terminator and we put the plant-based, tobacco-based extensor terminator that is good in polyadenylation and is efficient in termination and transit expression. And then we try different, we clone different promoters with Cas9, and then clone sgRNA and did some experiments with ICRs. Okay, so I'm sorry, I'll quickly go through these slides. We removed, like I said, the hygromycin and replaced it with NPT2 because hygromycin is a kind of there are some reports of regeneration problems and also some concerns. So we replaced with NPT2 first, and then we isolated this extension promoter from terminator, from tobacco. That's a smaller one, and we just clone it in this intermediate vector PGMTZ and sequenced 8. The same thing, we just cloned then. Different promoters, 35S enhanced promoter and PAO promoter, YAO promoter was also used, and ubiquitin 10 and ubiquitin 7 and intron. Interestingly, we could not get result with this ubiquitin 17 or intron. I think we were not successful in cloning these promoters, but the other one was okay. These are the source of this YAO and this ubiquitin 10 promoter was Arabidopsis that was amplified, and there were 1 kb and 650 base pair. And then we cloned just first, like before, in intermediate vector and then into the final expression vector. This is the sequence, just confirmation. And again, with the ubiquitin 10, this is its confirmation. And these ones, which I said that my student, he isolated from this potato, but we could not get good results. Well, you can say we were not successful in getting those, but the 35S, yes, fine. It was quite fine with 35S. And then the next, we inserted Cas9 just upstream to promoters. So this experiment just shows that we were just trying to see the efficiency of this extension terminator, whether it works or not. So we used it with Gus and with different promoter, of course, and the terminator as this extension one. And yes, the agroinfiltration results, they showed that it really works. So Gus fluorimetric assays, they also showed that, yeah, in this one, 2 double enhanced Gus extension terminator that showed the maximum Gus expression. And then guide RNA and this guide RNA is, of course, HPT, hygromycin phosphotransferase, the stretch that was, the Gus was interrupted with this stretch, and guide RNA was designed against this stretch. And then the sequence was confirmed. Sorry for. Okay, and then intrachromosomal recombination assay, and the guide RNA, they targeted this part, and then the Gus that got rejoined, we could see in our seedling. So this is
was one of the students who will continue the same work.
Internet is full.
Okay, my apologies to online participants. We are having some trouble here. Okay, so this cell wall invertase enzymatic activity is established that it's associated with pathogen infection in crops, and invertase inhibitor genes can have direct or indirect role in plant host defense mechanisms. So the one we were targeting, that was apoplastic invertase inhibitor, and this is just literature that shows that how this invertase inhibitor is involved in inhibiting the activity of cell wall invertase, and how it's suppressing its defense mechanisms. Again, some results, I think I put a table just to understand more. Yeah, for example, here you can see some altered invertase activity. They reported in beet severe curly top virus, and in impaired defense mechanism was a result was found in when they silenced this this cell wall invertase in tobacco, and again Xanthomonas oryza against Xanthomonas campestre. So the point I just want to raise that this invertase activity upregulated in Arabidopsis essential in modulating carbon partition and nourishment of pathogens, and the increased level of this one will lead to the ability of plant to resist the pathogens. And the inhibitor is what inhibitor does, it just inhibits the function of that vacuolar invertase. So we used a diploid and tetraploid potato for that purpose. And yes, first we characterized the gene. It's a different exon which we were going to target. The exon one is the generally the one which is targeted in literature. The first one, so we first cloned and characterized and learned its sequence, although its sequence was already available, just to confirm in our the diploid and tetraploid cultivars that were under study, and we found that, but with the diet, they all already reported that there are different alleles of this invertase inhibitor. So once the sequence was confirmed, we designed guide RNAs, and we cloned them in our target vector, but all this normal cloning procedures. The point I just want to make it is just we confirmed sequences. Again, then finally the expression vector were transferred to Agrobacterium. The point I just quickly want to make here, which we faced in our studies, maybe the people who are working on genome editing, they did not face. When we were trying to clone these our CRISPR constructs in E. coli, transfer our vector in E. coli, we could not get stable clones whenever we used JM109, DH5 alpha. So what resulted? We have to go for the XL10 gold strain that maintains the stability of these your constructs. So we were only able to get our clones in this one. And also we were really new. What we were trying to adopt a conventional cloning procedure, and always we were incubating our plates at 37. But what we learned, yes, by lowering the temperature at 28 and 30, you will get better colonies and more correct clones. And at 37, we were getting 1 or 2 or sometimes no colonies. So this is these are different explanation what can go wrong with this vector stability analysis. This is what we observed. Maybe the one people may not encounter with these problems. Yeah, then these are some results from from the calli. Some of the calli we just tested our constructs, knockouts in calli, and we cloned. Actually, these are the results of cloning, and this sequence shows the insertions, deletions, sorry. But interestingly, we also could not find our the gene edited one when the plants grew to a larger size. In some of the plants we could not find, but I mean at careless stage, yes, knockout was there, but when plants grew to a certain size and we checked again, the knockouts could not be confirmed. This is what also we encountered. And what we saw that also the Dr. Gosde, she also talked about, we tried different temperatures. And the 37 for the Cas9 to work efficiently, and 37 with the different and co-cultivation at 28 for 2 days. And we transfer intermittently to 37 while on calli induction medium. This was also good to get results, because we kept on changing temperature and giving at 37 centigrade, because the keeping your cultures for 37 for a couple of hours or times overnight may enhance the activity of Cas9. So these are different related stuff related to Cas activity. So the main point is the plants, we just check them, 0 and M6, this is deployed cultivated variety and the control, and also some of the disease lines compared to the still going on, and many more experiments are needed to confirm and establish the genome editing in these potato plants. Had examples for the moment, and I would like to acknowledge, of course, my previous university, Nido Morales Demery University, for providing us a good platform for the last 7 years to carry out our research work. And then my Center of Excellence in Microbiology in Lahore, where I just recently shifted. And then Erasmus, and in Erasmus, during Erasmus time, my student had a good work in James Hutton Institute with Dr. Abdullah Barakat. I'm really thankful, Dr. Emery Assay, in all this CRISPR-Cas work. He was the very active member. Professor Mehmet Chalishkan is a breeder, potato breeder, and always he was helpful in providing us the materials, and also teaching us how to keep this material grown in greenhouse and in field. So in CAM, Abdul Qayyum Rao, who is the PI of the work I mentioned about vacuolar invertase, and Professor Dr. Ahmad Ali Shahid Sahab, who is with us here and will present next lecture. With this, again, by taking advantage, I really want to thank Dr. Emre Assay and Dr. Shahid Mansoor for inviting us. It was great to be with you and to learn and discuss and interact with you all. Thank you so much.
Okay, thank you very much for this great presentation. Now we have 20 minutes to take a few questions from the physical participants. Are there any questions from the audience? Yes, can you come here? It will be great.
Thank you, Al-Baksh, for a very nice presentation. My question is that, one of the target gene, other than macular invertase. So you were saying that, in your slide, that probably you need to overexpress to have disease resistance. But then in your work, you were trying to knock it out.
Yeah. This one is cell wall invertase inhibitor. So this actually what happens, this inhibits post-translationally, this inhibits the activity of cell wall invertase. So what we wanted, we just wanted to knock down invertase inhibitor in order to increase the accumulated level of cell wall invertase. And the increased level of cell wall invertase means more resistance to appetite.
So have you confirmed the phenotype?
No, this is what is left. This is what is left.
Thank you very much.
Thank you.
You can ask for online.
Now we will look into the online participants if there are any questions in the chat. Just that. Okay, it looks like there are no questions in the chat. If there are any questions from the online audience, we can take them now. Okay, seems that I think there are no other questions online as well. Thank you very much, Doctor, for this great presentation. So, time, if you can finish. We can finish in 25 minutes. Okay. So basically, since we have finished this presentation slightly earlier than our program, therefore we would like to continue with our next speaker, and this will end our presentation session actually. Then we can give a lunch break. So our next speaker, Professor Ahmed Ali Shahid, is also coming from CMB, Center of Excellence in Molecular Biology in Pakistan. Dr. Shahid is the director of the center, and he is working on the regulations and public acceptance of transgenic plants. So today, I would like to invite Professor Shahid to talk about the regulation and public acceptance of genome-edited crops in the world. Yes. I will just prepare it. Share the screen. This is the pointers. Okay, good.
You can't it's full screen there.
If it make it full screen, then actually we lose everything. So you can just see it by using these buttons. It moves.
Okay.
And we see it actually full screen here.
Okay. Bismillah ar-Rahman ar-Rahim. First of all, I'm thankful to our giving me this opportunity to present something about the regulation and public acceptance of genome edited crops. So I am working as a professor in this Center of Excellence in 1984. So it has, we extend this. Before going to, I will tell some which are techniques which are being used for the modification of crops. So first is the crossbreeding. Crossbreeding, combining two sexual compatible species to create a variety with the traits of the parents. Then the mutagenesis, use of mutagens such as radioactivity to induce random mutations, creating the desired traits. Then the polyploidy is the multiplication of the number of chromosomes in a crop to impact its fertility. The next technique is being used as the protoplast fusion. Fusion of cells or the components to the transfer traits between the species. Then the next technique is transgenesis, addition of the genes from a species to create a new variety with desired traits. Then the genome editing is the techniques use of enzyme system to modify DNA directly with the core, with the cells. So then we focus in this conference on the gene editing. So how CRISPR works, is a general view. So it has the 4 steps. First is targeting. First we select the target. Then the binding process. Next is the cleaving process. And the final process is DNA repair. So in targeting, the scientists introduce the Cas9 guide RNA complete into a cell where it's randomly associates and dissociate with the DNA. Cas9 recognize and binds to a 3 nucleotides sequence motifs called ramp that is abundant throughout the genome. This is the first step. Then the next step is binding. In the binding, once it binds to a PAM motifs, Cas9 unwinds the DNA double helix. If the DNA at the location perfectly matches a sequence of about 20 nucleotides with the guide RNA, the DNA and the matching RNA will bind through complementary base pairings. The next step is cleaving. In the cleaving, DNA RNA pairing triggers the Cas9 to change its 3-dimensional structures and activates its nuclease activity. Cas9 cleaves both DNA stands at a site upstream of the PAM. In the cleaving, also the DNA RNA pairing triggers Cas9 and the changes its 3-dimensional structures again. Then the next step is repair. So in repair, it covers two steps. First is non-homologous ending joins, the other is homologous ending joins. A non-homologous ending join is more frequently used, faster repair mechanism because the cell does not use a template to join the broken DNA, and together it is however an error error-prone process that can introduce mutation in the target sequences. If the target sequence is within a gene's coding region, the mutation will likely inactivate the gene. In homologous directed repairs, the second type of repair mechanism is homology directed repairs, which is a less error-prone and uses a homologous DNA template to accurately repair the break. The scientists can manipulate this repair system by introducing the cell and access of a DNA repair template along the Cas9 guide RNA complex. Advantages of this technology. Offsite mutation can be avoided. Any gene can be targeted. In this technology, you can target genes. A row of improved traits have been introduced in different crops already. So there are so many crops which are being targeted by this technology, and they are giving good results, and they're improving their qualities and their production also. And the CRISPR case application, the selection of the targets. Yesterday there was a few presentations on this technique. So then after the selection, then the designing and synthesis of the sgRNA and promoters in plant binary expression vectors. The next step is the deliver vector to the plants. Then it has two steps. By the two steps, first is the agrobacterium transformation, then in the particle bombardment. So by using these techniques, which is available in your resources, you can use any technique to adopt this technology. Then we have the putative plants. The next step is screening of the putative transgenic plants. It also contains two steps. First is the PCR and restriction enzyme analysis. The other is the DNA, the plant that contain the next the generation advanced that contain the desired for that. So many importance. So the first is it effect on the agronomic values. So we can see in the wheat by using this technology, the wheat can be obtained by in the bigger grains that also increase the grain weight. If the grains are bigger, also it is automatically the weight will be increased. By this technique, the only female flowers can be produced to increase its yields. Then in canola, that increase shatter resistance, because this shattering is due to shattering there's big loss in the crops, so they can be increase shattering resistance. Then it also be useful in food and feed qualities. In the soybean, it can be obtained the improved oil quality, and in rice, reduce the arsenic contents by using the technology. And in wheat, which is a very important crop, then reduce the gluten content by using this technology. It can also be helpful in stress tolerance in the grapefruit and orange. In rice, wheat, and tomato, this can be of use to control the powdery mildew resistance. Then in grapevine, it can also be useful Then in the it can also be useful in the maize, wheat, against the drought tolerant, and in rice this can be used and the salt tolerance, and in soybean it can be helpful drought and salt tolerance. Then it's also be increase the fiber quality of the wheat, and also drought tolerant maize can also be produced, and also in the potato with reduced PPO and glycolides contents. Then in wheat also mildew resistance wheat can also be produced by using this technology. Then it is also helpful in the food security, and then climate crisis it can be used helpful. Then biodiversity conservations, and then the dependence on the heavy inputs and fertilizers, pesticides, and irrigations, we can minimize these things. Then this there are four techniques which we can use. We can edit the gene. The first is the ZNFS, then the TALENs, then MN, and then the CRISPR. So this techniques have the four categories, and the first category is SDN1, and the second category is SDN2, and third category is the SND3. So in SDN1 application mutation consisting of the changes in a few base pairs, and the short deletion or insertions are generated in the binding regions in the genome as a result of the error-prone gene repair mechanisms of the cell. In SDN2 application, specific point mutations, small deletion additions are generated as a result of the introduction into the cell of the repaired DNA templates. In SDN3 application, then the process versus product-based GMO regulatory frameworks. So some people believe on process-based technology, and others believe on the product-based technology. In process-based, they consider the techniques used to create new crops, plant varieties. So they take inquiry about which process was used for these techniques, then regulated if nucleotide acids are deployed during the development of a crop. And in product-based, they focused on the final product, no matter how it was developed, based on the potential risk of the new trait or attributes introducing into the plant to a public or environment. So this is the slide showing the landscape of the regulations. So there are different countries are adopting different techniques. Then in Canada is considering the product-based technology, and in the USA they are also opinion about the product-based technology. Then in other Colombia and Brazil, they are product-based technology. They believe on that. And other countries like European Union, they believe on the process-based technology. And Russia still to decide. They did not decide. Still, they are thinking about that which technology they should adopt. And Australia, they believe on the process-based technology. In New Zealand, they also process-based technology. They are adopting this. This slide shows the Nbt Assessment Roadmap. So there are a few steps in that. First is the biosafety committees. Then there a new combination of the genetic material. So if they say no, and other side, then they have, does the Nbt use a transgene temporarily? Then if they say yes, then that is a GMO. And then next is does the Nbt use a transgene technology? If yes, then is the final product free of the transgenesis? then they do more things about that. And if they say no, then they say GMO. If they said yes, then they go to the So there are different countries showing Argentina, Brazil, Chile.
Okay, we can continue.
So now we will talk about USA. The USA does not regulate or have any plans to regulate plants which could otherwise have been developed through the traditional breeding techniques, as long as they are not plant pest or developed using the plant pests. So they have the website MI regulated under 7 CFR parts 340. Then, but deregulated organisms by the USDA APHIS may still be subject to other regulatory authorities. In Israel, Ministry of Agriculture and Rural Development, National Committee for Transgenic Plants, then the gene editing, there is some that the Commission reconfirms its previous decision dated that plants which are the progeny of plants that underwent targeted mutagenesis utilizing gene editing methodology that caused the deletion of nucleotides and were proven that there was no insertion or incorporation of the foreign DNA into the genome of the plants do not fall in the category of transgenic plants. Therefore, plants, their products of this technology will not be considered transgenic. In Australia, new decision 10 April in charge, then the no regulation for the use of gene editing techniques in plants, animals, and human cell lines. They do not introduce new genetic materials. Valid for the use of tools in which protein cut DNA at specific target sites, as long as the tools allow the host cell to repair the break naturally rather than using a template containing genetic material to direct the repair process. In Russia is embarrassing gene editing's federal programs wants to produce 30 gene-edited crops and animals by 2027. Some gene-edited products will be exempted from a law passed in 2016 prohibiting the cultivation of the genetically modified organism in Russia. New decree describes gene editing technologies which do not necessarily insert foreign DNA as equivalent to the conventional breeding method. In Europe, then old GMO regulation, but discussions are ongoing. EC takes a new attempt to assess NPBTs by April 2001 to start a science-based decision process already started. So they define the GMO means an organism in which the genetic material has been altered in a way that does not occur naturally by mating or natural recombinations. So this slide shows the current status of EU legislation on crop breeding. So we can see here, though the mutagenesis, they consider it safe, although it has so many things which in the mutagenesis takes place, and there are so many steps. And also in the crosses, there are also mutagenesis, other ways technology, but they considered that it is safe. But in CRISPR case technology, where is only gene editing is used, so they are thinking that it is dangerous, and they are very reluctant to adopt the technology. So we have to think of what are the behind these things, why they don't accept this technology. Well, actually, there are so many differences. The other technology are very complicated, but this is very simple technology, or they will not accept these things. Then these are the policy developments in different countries around the world. So we can see from this slide that the green things is positive decisions in these countries. They are taking the positive decisions. They are thinking to accept this technology. And then in the yellow, there is a no farmer decision on these countries. And in the red, whereas red is showing that is the restriction decision. So we can see that in Canada, product-based approach, ongoing discussion to improve the systems. So they believe on product approach, but there are always chances to improve this technology. Then in US, proposed new USDA policy excludes certain techniques, executive order to modernizing the regulatory framework for the biotechnology. In Argentina and Chile, Brazil, so they case by case, the certain in Europe, the decision cells mutagenesis GMO and the subject at EU GMO regulations. In Russia, we can see that the cares are saying that conventional like as GMO. In China, proposed. In Israel, outside the GMO scopes. In Australia, revised gene technology. And in the New Zealand High Court decision that few specific techniques are GMO. In our country, there is also government has the case by case, but they will first check the case on which crop the technique is used, then they will decide can they adopt it or not. Then this slide shows the study presenting experimental field trial data. So in this we can see in USA they are adopting the tomatoes. Then Sweden potato, and England also they are using cauliflower. In China tomato, and Japan they are also adopting to producing the tomatoes. So this is the last slide. This we can conclude from this presentation that publication on genome editing applications are increasing continuously worldwide. Then CRISPR's case is the dominating tool that is being used, and with passage of time, the people will be used to adopt this technology. And in the beginning, there are some problems, but ultimately the people will come to adopt this technology. Most applications that are 90% introduce targeted mutations. Only 10% insert heritables, that is foreign DNA. The China and USA are the leading countries to adopt this technology. Then application address a huge diversity of different characteristics addressing all kinds of relevant agriculture or consumer traits. Then more and more countries follow a differentiated regulatory approach, taking into the account the kind of genetic change. And this is about the conclusion. Then at the end, I will acknowledge the scientists who are working in area of the genome editing for their continuous efforts towards food and nutritional security. Then all researchers who publish their works in peer-reviewed journals on genome editing. So thank you very much for your attention.
Thank you very much for the presentation. Do we have any questions from the audience? Can you come closer to the
Thank you for your presentation. I'm sure that most of the people sit down here, that's what now going in the climate change is by handmade of the man. So my question, in natural, that's not happen that of maize can cross wheat, that's an example just. So foreign DNA in a certain crop, what's the effect or other consequence on the wild progenitor, especially this wild progenitor will stand for a decade. So what the effect when we started to plant any crop and have a foreign DNA and plant it in the field. There's an study or expectation in the effect and the consequence in the wild progenitor that in fact this is the storage DNA and will stand for a decade and maybe losses maybe but there's any expectation for this?
Yes, because we are playing with the nature, because nature has a set process. When we insert or delete any DNA or changes, we are expecting that there should be, we expect that maybe they will go beyond their natural process. So to check this technology, they have, though people are worried about that, maybe the basic thing may be changed with that.
But gene editing, it's not a nature.
Yes.
So maybe we lost the wild type progenitor. How can keep it for the next generation, not for our generation, for the next generation, how can keep it?
So we should continue practicing this, and we should closely check every steps. If there is any change, then we can use the technology. We can do this.
Can we control this? Can you control, in fact, in the world, can you control the distribution with field with Fulgi and foreign DNA?
Yes, up to extent we can.
And not to extent, I sit here and we talk about in general, not in Jordan, not in Pakistan.
Yes.
Here taken in the future for a generation, not for us.
Yes.
So how can keep save it?
So because it can be safe because the population is increasing day by day, and we have to adopt this technology. Otherwise, there will be no food. If and there are so many diseases and there are so many threats to the class. So we have to accept this technology. Otherwise, we will have nothing to eat.
So you are saying destroy the future?
To save the future.
To save, no, that's not save the future. If you are lost the wild originators, right? Okay, thank you.
That's okay. Yes, that's it.
I think that there are many people that can answer the question, but just to be for general audience, obviously, this is a question that has asked many times to people like us. So there is a safe history of use of genetic engineering. We have been using transgenic cotton, maize, canola for a long time, and so there's a safe history of safe use. Now, whether to plant it in a country or not, that's a political decision. The government make a decision decision whether to. So there are countries that allow you to plant GMO, and countries that, support either because of the trade or because of some other, unknown fear. So I think that, but the history is very much there. Arsenic, cotton, for 20 years seen any adverse effect. Safer because it's just like having is a dilemma that EU for creating mutation, and it is just like happening because you select the best mutant. So this is something which is acceptable to the whole world, but genome editing is relatively, I would say, is considered as safer because in most of the cases there is no transgene. So most of the countries have adopted a very practical approach. I was very pleased with the very practical approach of Japanese and Australia. So I'm sure that there are gene banks available with thousands and thousands of lines of every crop species. If you are not interested to grow, and if you want to remove one of the edited crops, you can always do this. In agriculture, we are replacing varieties very quickly. So especially in case of wheat, the varieties are replaced after every few years. So I mean, this is an ongoing process that you select the best, and if it is not working well, then you can replace it. So if there's no that the GMO will stay always, or edited crop will as long as you have some benefit, you can grow them, and then if you are not very happy with, for example, you can replace it with another variety. Remember the comments by Dr. Mark Tester. He said, I mean, the important is that we want to have food security. We are, and genome editing is one of the technology. Dr. Mark and Dr. Haitha has really very well explained that that how important it is that to mutate all the 3 genomes. It is something which is not feasible a few years ago. Is a remarkable development in the history of mankind that we are able to do very quick and very specific mutations. I must say that so this happens whenever you bring any new technology. Even there are people who are against vaccines. People who say we don't want our kids to be vaccinated. So this is always a choice. So this is the society as a whole decides that whether you want to adopt or not adopt our technology. We are here sitting here. This meeting, it was possible because all of us are vaccinated. So this is a remarkable achievement of mankind that within short time we were able to come up with vaccines, and so there are people even in the developed world that who have doubts and they don't want. So this is a free choice that you have, that whether you want or not. So I think that, sorry for a long comment, but I think that it's very important that we keep the science progressing and adopt whatever methods are available, and as a society decide whether they want to adopt or not. There are some of the I mean, when we were starting our biotechnology program, there was so many ideas that could not see the light because they were not. I mean, they remain an idea, but there are others who have gone into the field and created an impact. So so we'll see. So much we'll continue to make process and then make choices. Thank you. Sorry. Dr. Haida.
I think you give me, I can ask, because I don't know the regulations in Turkey and Pakistan. It is that now hosting countries to organize this workshop. So could you tell me what is the regulations in Turkey and Pakistan? And also second stuff, is there any strategy to promote the genome editing in Turkey and Pakistan, like outreach activities or our awareness, the publicity, this kind of activities, there any activities in the countries?
Sure. I can tell about.
Thank you for the explanation. That's great. Thank you.
I can tell about Pakistan. So there's a regulatory authority. It's called National Biosafety Committee. There's a technical advisory committee. I co-chair that advisory committee in Pakistan. And in 2019 we held the meeting, and we, as a country, we welcomed the technology, and we said that we will do. You still have to apply for biosafety clearance, because as Dr. Ahmed Ali Shahid has told that there are three possibilities that you can also have a transgene when you are doing theoretically. This mostly it is knockdown. So the policy is that we will discuss the applications, and most likely we will declare whether the technology is going to lead to the in the final product, whether there's a transgene involved or not. If there's no transgene, if it's just a mutation, which will be considered as non-GMO or freedom to operate. So this is the national policy of Pakistan, and we are promoting this technology. So maybe Emre can tell about the Turkey, but I'm sure that probably similar situation in Turkey.
Now, basically, the in Turkey, we are following the EU regulations. So, and we just need to see their decision on genetically modified gene-edited crops. But other than that, for the general GMOs, it is highly protected, regulated. We can produce them. We can establish the lines in the laboratory. We can take them to the controlled greenhouse conditions, but we cannot take them out. It is illegal from so all the seeds we import, they are highly regulated against this GMO. From the ministry, they may have some extra things, but not this much. If there are anything change.
A part of the for we signed the protocol. That's why we should follow the protocol. But now, from now, we are not allowed to anything try it. We just allow to use the GMO products for feed production, not feed production, just for feed usage. We import the seeds or some events from. We are importing some seeds for just feed usage, not anything else, and also some for painting industry. I think that's all. We are not allowing as for now. And it is the same. There isn't any difference for the yeah. There isn't any difference.
We have some comments at the back. Yes, you can come.
Actually, we always talk about the sites that we always, in terms of consuming GMO products. What about only seed production? I mean, those countries, do they allow
seed production in their own land, in their own countries. For example, European Union, there are many countries they invest on GMO and gene editing, and at some point they have to actually test this on some field, and then maybe under different environmental conditions they want to select different lands, different countries. What is the actually legislation about this? For example, in Turkey and other countries that are restricted against, they restrict using GMO as food group.
I will tell about Pakistan. In Pakistan, there is a, if you have the transgenic or any GM crops, so you can grow in your country. There are different legislation bodies are there, passing through them. There are some rules, regulations, then you can grow in your country. But we cannot produce, you export to other countries. But I don't know about Turkey and other countries, because each country has its own regulations. So they adopt their own regulations. But if they export a country, there are international regulations. So they follow that regulations.
I can add some. So it also depends on the trade. For example, we allowed rights for transgenic maize in Pakistan for 3 years, and then the case was finally good for the commercial application, commercialization. The field trial is allowed. The commercialization was discussed, but the trade that were offered were, insecticides and herbicides. And the overall feeling was that these are not really the trades that are required by the farmers, because the situation is changing. Even in maize, so cotton is grown. 95% of cotton is produced in Pakistan. For maize, we said, okay, for the time being, there's not much benefit to the farmers. But with the rise of fall armyworm, we are thinking to reconsider cultivation of maize in Pakistan, because there's a big problem. So 2 years ago, there was no fall armyworm in the field. So we didn't see any benefit, and also the other problem with the maize was that the pollen travels a long distance, whereas non-GM so, but the thing depends ultimately on the trade and the benefit of the farmers. The farmers have the benefits are good. Thank you.
I just know one case for Turkey, and I remember late 90s or early 2000 in Turkey there were some transgenic corn trials in Adana part, and then I researched what I have seen in different conferences. People talk different about the results. Some people, actually the results were conducted under Ministry of Agriculture. So some people show a data where the transgenic corn, the yield was almost double. And but some people don't believe those results. So it's a kind of a debate still going on. But yes, it was a foreign company that somehow conducted their trials in the country. But after then, there was, you can say, there was no any trial. This is what I know conducted in Turkey about the transgenic. And yes, this is the case. And the earlier question, I just want to add, I got an opportunity. The people who like Gmo editing who are hopeful. In EU, there is a consortium called SAGE. This is a consortium of 137 countries. achieved that definition, and they recently, when she was one of them, Imam, they published a very good article and defining how sustainable and how we can do that. We're not in you also from ministry. If something changes in you, definitely it will impact many other countries, including us.
Also, I think UK has different.
Yeah. Because Brexit, because we didn't need it now. So basically, it's kind of our research is not that Gmo Regulations. So now, as we can do Protoplasts, especially the genome editing process for research purposes. So it is great development for us. Also in UK, there is a kind of promotion of genome editing, kind of for public awareness or the toxicity or outage activities, and it works. Now farmers are more open to genome editing process, and they are able to use it, but they are just considering how they are going to find money. Because if they can sell their products in Europe, they can make money. That's the only problem. But obviously, I don't think UK wants to is the opportunity to use genome editing for breeding, because it's shortened period and can distinguish from the normal mutation rate. With the mutation rate, I told you, 500 for mutation mutation, and we can't increase it, just upgrade. And they don't want to use this opportunity because if we just do research, we are advanced, we put this, but it can come to market, but probably not in this case. So they don't want to use this opportunity. So we are open with that, but just we are ready to limit because for us, the market, second start, and also now we can use it for research purposes, but eventually in future is going to be more open.
Thank you very much. Because I have some small comment. I have been working with transgenic plants a long time, and several transgenic plants, including rice and other processes. We observed unexpected metabolite changes, not on all of them, but some of them, especially transgenic plants generated by tDNA mutagenesis, sometimes made very hazardous. Some of them were observed very strong accumulation of thermolytic compounds under hot stress, which can be accumulated on the seeds and then they cause allergic reactions. So in that case, I think we should not go only for the new technologies. Also, we should improve all technologies in the midst of the making our products better, especially for food business. We should be very careful. Thank you very much.
Well, this is very interesting because in literature, if you look at different articles, except papers from Chavini group in France, there is no, concrete kind of evidence. I want to introduce the word substantial equivalence. If your transgenic crop plan was not substantially equivalent with your control, yes, definitely you can say that there might be some pleiotropic effect of your introduced backbone genes. That's quite possible. But the beauty with the working with the plant is that you can discard your plant if the phenotype is different. You just discard, and there are regulatory mechanisms, frameworks. What we learned from this lecture, that is the main mandate of those regulatory frameworks, if to assess all these genomic data, GMOs plants at every step. And once all the parameters are completed, then they are allowed for the next step. So all things are in consideration.
Anything from Mark? Yeah. We actually did a comparison of transgenics and non-transgenics
and plant-produced through tissue culture. And we actually found that there was more changes in plants which were produced through tissue culture, no changes in metabolites than what were in the transgenic or transgenic plants. So tissue culture is quite a stressful environment for the plants to regenerate. So we actually found more changes through the tissue culture transgenics. So I think every transgenic plant or event needs to be assessed on its own merits, and it really depends on the position that the TDN has gone into the plant, and also the gene that you're regulating down regulating. This kind of thing.
I just want to add something. I think the technique is not the problem, but the thing is the dynamic of genetics. We should know the dynamics of genetics and then how one gene affects the other, how one gene affects the other gene affects. So we have to think more, on a macro scale, and then we have to see the effect of that phenotype. That mutation, and then which phenotypes are affected simultaneously. So it's not actually related with just gene editing, the CRISPR methodology. Yeah, you can do this with monoclonal mutation under the tissue culture environment. So it also happens in nature. Mutation. So you can do, you should be aware of your
Let me see. Okay, in the chat, we don't have any questions from the online participants. Now it is 12:30 in Turkey, and we will give 2 hours of break for lunch. And afterwards, we will come back for the online presentations of some posters. We will have, I think, around 14 posters. We'll have 5 minutes. And right after that, we will finish all the lectures. Thank you very much. Thank you.